cord. There we go. All right, thank you. Has everyone gotten a sign-in sheet? I always have it up front, you know that. So I wanted to preface this with, this is the hardest chapter thus far. So I'm not saying that to scare you, but I want you to be prepared for the requisite amount of study. So I know the bones was our first chapter, really getting into it. This really gets into it. Not the video. What's that? You can't see me? As long as you can see the screen. Study. So you can absolutely do it. I'm recording this. I'll put it on my YouTube page so you can review. Okay? But we're getting down to business. So let's lecture for like an hour, then we'll take a break. I'll give you the exams back, and then we'll pick up where we left off. All right. How does it look on the screen here? You can see it okay, right? Okay. All right. So this first chapter is muscle tissue. Much like we had for the bones. So remember we had one chapter on bone tissue, and it talked about the structure of the bone, how bone grows, how it repairs, yada, yada, yada. And then we had a second chapter on the bones themselves. It's the same here. This chapter is muscle tissue. What is the structure like? How does it contract? And then the next chapter is just the muscles themselves. Cool? Daisy was like, put your butt down in, Christy. Put your butt out. I'm glad I could be a source of laughter. I'm terrible. So I want to remind you, in the tissue chapter, we talked about 3 types of muscle tissue: skeletal, cardiac, and smooth, correct? This is just a review. Look, we're starting off easy. Okay? So I just want to refresh your memory. Skeletal muscle is striated, correct? Voluntarily controlled. These are the muscles that are attached to bone. Tonight's lecture will be focusing on skeletal muscle. Don't forget, we also have cardiac muscle, also striated, involuntarily controlled. It's a little bit more spread out. Cardiac muscle has those intercalated discs. Every so often there's a darker band. Is this coming back to you? And then smooth muscle, of course, does not have striations found in the walls of organs, helps to push things out, like a baby. Involuntarily controlled. So just review that. We're going to be focusing on skeletal muscle. If you have questions, you're going to ask, right? Now listen, all muscle tissue has some unique properties. This is our first time talking about this. We talked about bone. That didn't really have these same unique properties. So this is a first. I have put stars beside the ones you need to know. These are some characteristics that are unique to muscle. And they're unique to all types of muscles: skeletal, cardiac, and smooth. It shouldn't be that surprising. Can't muscle contract? If we send a stimulus into it, it can contract. And after it contracts, it is elastic, so it can contract and then it can return to its original resting position. So in that way, it's elastic. It can contract and shorten and then return to its original length. It is also what we call excitable. It's responsive to stimuli, The stimuli is a nerve impulse. When we send a nerve impulse into a muscle, it will contract it. Not every type of tissue is responsive in that way. If you send a nerve impulse into a skin cell, it's not going to do anything. So these are unique to muscle. What do you have? Is your ears?
Yeah.
They're my ears. I had to take them off because of my head.
One's big and one's small?
No.
Oh, it's just flat down.
So. Solid.
You're special, Seth.
Where are your glasses?
I've been on a few shows.
Hey now. All right, so these are characteristics that are unique to muscle tissue. It makes muscle tissue what it is. Good. Now, here's where we get to some of the nitty-gritty. Okay, we're going to spend the majority of this chapter talking about skeletal muscle. Of the three, skeletal, cardiac, and smooth, we're going to talk about skeletal the most. That's because we have the most skeletal muscle. How many skeletal muscles do we have? We have over 600 skeletal muscles, which is to say a lot. So we're going to focus our discussion on skeletal muscle. At the end of the chapter, we'll come back around and I'll say, well, here's how cardiac is a little bit different. Here's how smooth muscle is a little bit different. So we're going to focus right now on skeletal. You with me, skeletal? As it turns out, skeletal and cardiac muscle are very similar. Cardiac and skeletal are very similar. Smooth is a little bit more different. Okay, so if we are to look at a skeletal muscle, let's start big and break it down into smaller and smaller pieces. This is just memorization, folks. Kind of like the bone, you know, there was the diaphysis and the epiphysis. The osteon was in the diaphysis. The osteon had the lamellae, the canaliculi, the osteocytes, the Haversian canal. Remember all that beautiful stuff? Memorization. Be careful though, because the words kind of sound alike. Let's start big. We have the whole muscle. The whole skeletal muscle is made up of these rounder sections called fascicles. So each one of these is a fascicle. Here's a fascicle. This circle is a fascicle. This circle is a fascicle. Each fascicle is a collection of muscle fibers. So this whole circle is the fascicle, but the fascicle itself is made up of individual muscle fibers. Each muscle fiber, and now we're getting super microscopic, each muscle fiber is made up of smaller components called myofibrils. And the myofibrils are made up of the smallest pieces called myofilaments. I'll get the smile on screen. I don't know. I think it's easiest to start big and work your way down. Some people might like to start small and work your way up. Big skeletal muscle is made up of these round segments called fascicles. Each fascicle is made up of muscle fibers. Here's a muscle fiber, here's a muscle fiber, here's a muscle fiber. Each muscle fiber is made up of myofibril, And each myofibril is made up of the smallest, which are the myofilaments. Aren't you loving the muscles already? Mm-hmm. Some important things to point out. These myofilaments, of course, they're the smallest. But what's really important about the myofilaments is they are the contractile. When we talk about a muscle contracting, what's actually contracting? That was for emphasis. The myofilaments. So the myofilaments, those smaller pieces, that's what contracts. My goddaughters don't find me nearly as funny as you do. They just look at me. I said, I bought your asses Hamilton tickets. They're not cheap.
Do you need Michael?
That's why I work on Saturdays, right? Work hard, play hard. Once in a while. Most of the time I'm just here. So, we can see these. I would never ask you this on a test or anything, but, this is a whole muscle. A whole muscle is made up of fascicles. Here's a fascicle. Each fascicle is made up of muscle fibers. Muscle fiber, muscle fiber, muscle fiber. Hello. Some other things to point out. You only have to know here what has a star in front of it, the epimysium. The epimysium is the connective tissue covering over the whole muscle. The epimysium is the connective tissue covering over the whole muscle. Epimysium, some people say epimysium, I say epimysium. You saw this in the cat dissection, didn't you? Didn't you see that thin connective tissue covering the whole muscle? What you were seeing there was the epimysium. There are other connective tissue coverings. For my class, you don't need to know those. You only need to know the covering over the whole darn muscle, epimysium. Not so bad, are we? The next couple of slides go into more detail about that order. Just going to keep repeating it again and again. We have the whole entire muscle, and the whole entire skeletal muscle is made up of these smaller circles called fascicles. Each fascicle is made up of muscle fibers. By the way, when we say muscle cell, we tend to be talking about the muscle fibers. Not a big deal, but each muscle fiber, so if we take this one muscle fiber and zoom in, each muscle fiber is made up of myofibrils. And then each myofibril is made up of the myofilaments. Okay. I don't know about you, I kind of like this picture. I like seeing it all together. All right, let's talk about those myofilaments. Smallest ones, what's important about the myofilaments? They're contractile. There are two different myofilaments, actin and myosin. Maybe you've heard of these before? Yes? If I go back for a hot second, these are the two, right? Actin and myosin. So the slide I'm about to show you is showing these same things. The two myofilaments are actin and myosin. Actin is the thinner one. You should know that. I remember that because actors are supposed to be skinny or thin. That's how I remember. Actin is the thin one. Myosin is the thicker one. So of the myofilaments, the contractile, smallest pieces of a muscle, actin is the thin one, myosin is the thick one. Actin tends to look like this. It's kind of two pieces braided together. Actin has these two subunits. I don't need you to memorize them from the picture, but for right now, I need you to know that actin has these two subunits. I'm not going to ask you to label them, but troponin and tropomyosin are subunits on actin. Troponin and tropomyosin are subunits on actin. They can be spelled with either an I or an O. You'll see either one. Troponin and tropomyosin are subunits on actin. Anybody here work in a cardiac unit? Do you do troponin levels? Troponin. So in what instance, well, I just gave it away, didn't I? I asked what unit you're on. And what type of, what body system could be going wrong if we're measuring troponin levels? The heart, right? So even though we're focusing on skeletal muscle, cardiac muscle is very similar. So if someone has had a heart attack, some of the cardiac muscle tissue is dead or has broken down, and you will see remnants of that broken down cardiac muscle tissue, troponin. So when troponin levels go up, that's a bad thing. Troponin should be staying where it belongs.
When skeletal muscle breaks down, does it also release troponin?
It does, but it doesn't tend to in the same way. It does a little bit, but doesn't in the same way. There's other markers that we would look for more in the skeletal muscle, like myoglobin, creatine phosphate, things like that. Okay, so actin and myosin are the myofilaments, smallest pieces, contractile. Actin is the thin one. Actin has these two subunits called troponin, tropomyosin. Myosin is the thick one. And what's important to note about the structure is little heads and little tails. Heads and tails. This is our first chapter where we're going to really get into some physiology. My Hudson Valley ID has a little button that says, I love physiology. So my advice to you is I encourage you to first learn the anatomy, right? So I know we're going to go through it all tonight, but when you sit down to study, you can't study it all at once. So I encourage you to start with the anatomy. It'll be much easier to figure out how they work together when you actually recognize the anatomy. Okay, now, these actin and myosin, they arrange themselves in a unique pattern. Do you see at the top? The actin and myosin, they kind of alternate, don't they? Red is the myosin thick, blue is the actin thin. Don't they kind of alternate? So it's important to point out how these actin and myosin are arranged. they're arranged in these horizontal units. You're looking at one of these horizontal units. So once we can identify what actin and myosin are, the next step would be to recognize that they exist in a pattern, like in a horizontal unit, where they alternate. Can you see what I'm saying? The actin and myosin are arranged in this horizontal pattern, horizontal unit. That unit is called a sarcomere. Losing it. Sarcomere. That horizontal unit is called a sarcomere. So a sarcomere is a horizontal unit of actin and myosin. A sarcomere is a horizontal unit of actin and myosin. These are both pictures of the same thing, pretty much. So these actin and myosin are arranged in these horizontal units called sarcomeres. So look up here. This is actually part of the myofibril. So if we zoom in on the myofibril, remember, the whole muscle, fascicle, the muscle fiber, the myofibril. It is the myofibril that is made up of the myofilaments. So if we look at this myofibril, it is going to be made up of the myofilaments. So here we've zoomed in, and we can see that actin and myosin arranged in a horizontal unit. The blue are the actin, the red are the myosin. Isn't it alternating? So we have one unit of actin and myosin next to another, next to another. So the myofibril ends up being one little unit, one sarcomere, next to another, next to another. I just showed you what one little sarcomere looks like, right? But of course, it's not just one. The actin and myosin form one unit here, one sarcomere, and then right next to it's another, and another, and another. Because what's going to happen is, each individual sarcomere will shorten. What we're going to find is how muscles contract. It's called the sliding filament theory. So at rest, the actin and myosin are, like it shows here, they're alternating, but they're a little bit apart from each other. When the muscle contracts, the actin and myosin slide together. So what we're going to find is, when one sarcomere shortens, then the next one shortens, and the next one shortens, and the next one shortens, then the whole darn muscle shortens. But more of that to come. Aren't you glad you're here today? For now, we're just identifying the structure. The actin and myosin are the myofilaments. They're arranged in a horizontal unit called a sarcomere. And it's just one sarcomere next to another, next to another. These are the contractile parts of a muscle. sarcomere, it's possible to label the A band, the H zone, the I band. My colleagues ask students to know these, but I don't. This is a rare instance where I cut something out but they don't. So you don't have to know these definitions. Is that okay with you? They're just a way to, I mean, who cares? You don't have to know it. Why should I even say anything else about it? Even the start one, you don't need to know. I've trained you well. Grasshopper. Now, this is the image of the sarcomere, the unit of actin and myosin. This is how we draw it to understand it. But like you saw in the microscope in the tissue labs, do things always look in real life like they look in the picture? No. So we use these pictures to help us understand it, but under a microscope, it looks like this. We get the idea, though, that there's alternating bands of thick myosin, thick and thin actin. The myosin has little heads. That's pretty cool. So I've already begun to introduce this. This is what a muscle at rest looks like. So here's our unit of actin and myosin. At rest, do you see this space right here? So when a muscle is at rest, the myofilaments have not slid past each other. They're apart. And then the bottom, when we contract, again, the myofilaments slide together. So in the top, there's a space here between the ends of the actin, because the muscle is relaxed. When a muscle contracts, the actin and myosin slide past each other. They slide together. More to come, but I'm just wetting your whistle. I'm going to show this video to my goddaughters. They're going to hear the laughter. And Christine is funny. My youngest goddaughter, the 15-year-old, missed her school bus this morning. And Christine drove home from Maryland, got here at 11 o'clock last night. I was at work at 7:30. She's going to have to learn. We can only go out and do things if you get up for school. The moms out there, do you hear me? Just trying to pause to let this sit. Okay. There's a couple extra pieces of anatomy, and then we're going to be done with the anatomy. So check it out. There's this sleeve over the myofibril called the sarcoplasmic reticulum. It's this bluish purple thing. It's not a complete sleeve. It's kind of like porous, but it's like a network of tubes. The important thing is, it's a sleeve that covers the myofibril called the sarcoplasmic reticulum. Its function is to store calcium. The sarcoplasmic reticulum is a sleeve around the myofibril. Its job is to store calcium. We're going to need that calcium for contraction. I'm just introducing all the key pieces. Sarcoplasmic reticulum, it's that covers the myofibril stores calcium. Blue. The blue thing. All right. There's much more, but those are the basics. So when you go to study, I encourage you again to learn the anatomy first. Then it'll make this next part much easier. Okay. You with me? Reasonably enough? Because now we're going to start to move into how these parts work together. The overall idea of how a skeletal muscle and cardiac, for that matter, how it contracts is the sliding filament theory. These myofilaments, actin and myosin, slide past each other. When all at each sarcomere, if they all slide together, then the overall muscle shortens. And because our skeletal muscles attach to bone, when this shortens, what happens? It pulls up on a bone and it creates movement. Okay. Now, we're going to lay out how this happens, but first, in order for a muscle to contract, it has to get a message to contract. What is the stimulation that tells a muscle to contract? Nerve impulse. So that's where we're going to start. The very first thing, before we even get down to the structure of the muscle, the very first thing that has to happen for muscle contraction, there has to be a nerve impulse that goes to the muscle to tell it to contract. And that's true no matter if it's skeletal muscle, but also cardiac muscle. Just because cardiac muscle is involuntary doesn't mean that there's no nerve impulse. The brain is constantly sending nerve impulses to the brain, to the heart. The brain is constantly sending nerve impulses to the heart. It's just involuntary and it's being done by your brainstem. Versus skeletal muscle that is voluntary, correct? Yes. So that's the first point. The very first thing that has to happen is we're going to talk about how a muscle contracts. The very first thing that has to happen is nerve impulse has to go to the muscle. That's the stimulus. How does this happen? Well, that nerve impulse leaves the brain, goes down the spinal cord, comes out, and the nerve, here's the nerve ending, that axon, the nerve ending comes down to the muscle. You may recall, you may know, if we actually zoom in, when a nerve goes to a certain part of the body, it doesn't actually touch that, right? There's a space called a synapse. So here we are, we're zooming in on where the nerve, the yellow thing, where the nerve comes down to the muscle. Here we zoom in on that, and we see that between the end of the nerve and the muscle is a space. That space is called the synapse. You've heard that word before, haven't you? This junction is called the neuromuscular junction because it's the junction between the nerve and the muscle. Neuromuscular junction. Junction between the nerve and the muscle. impulse into the muscle, which is the stimulus, which tells you to go. Do you notice there that the axon of the nerve comes down, but then it has like little tendrils? So the nerve ending has little tendrils that wrap around the muscle. That's efficient, isn't it? So we can actually send nerve impulses down each little tendril. There's a picture of this later. Let me show you. It's kind of cool. Look. Here's a real picture of that muscle tissue. Here's the axon coming down, and each of these branches is like a little tendril that wraps right around the muscle fiber. That's kind of cool. Some people say. But some people don't. But for your exam in 2 weeks, you're going to say it's cool. Okay, so here we see a slight issue because we initiate a nerve impulse from the brain. It's going to come down to the muscle, and that nerve impulse is going to tell the muscle to contract. But wait, when that nerve impulse gets to the end of the axon, it reaches a dead end, doesn't it? Because there's a synapse here. The nerve doesn't actually touch the muscle. So how are we going to transmit that electrical current? How are we going to transmit that nerve impulse across the synapse? I didn't hear you. I'll tell you. So sometimes when I'm trying to relay this physiology, I find it helpful to do it in steps. Because oftentimes this is how it goes in the body. This happens, and then this happens, and it's like a domino effect. So here's our first example of that here in AMP1. How is that nerve impulse, that electrical current, how is it going to get across the synapse? Well, here we go, ladies and gentlemen. First, that nerve impulse comes down to the end of the axon. Nerve impulse comes down to the end of the axon. Nerve impulse reaches the end of the axon. When that nerve impulse reaches the end of the axon, calcium channels open and calcium comes in. When that nerve impulse gets down to the end of the axon, that electrical current triggers calcium channels to open and calcium comes in. If there's no electrical impulse, calcium doesn't come in. So nerve impulse reaches the end of the axon. That triggers calcium channels to open. Calcium comes in. When calcium comes in, it's going to trigger the release of a neurotransmitter. Sorry. So the answer to how we deliver that current across the synapse is through the use of a neurotransmitter. These green dots that cross the synapse, they are neurotransmitters. Because I don't have my iPad to write on, let's do this. So it's a neurotransmitter. These are chemicals that cross synapses. So how do we actually deliver, how do we transmit that electrical current across the synapse? through a chemical messenger called a neurotransmitter. Doesn't the name kind of sound familiar? A neurotransmitter? It transmits something from the nerve. We see these released from all nerve endings, everywhere in your body. This is the only way that we can deliver a message or transmit that nerve impulse across the synapse. You've heard of a bunch of these. I'm not going to ask you all of these. Haven't you heard of dopamine, serotonin? Have you heard of GABA? Have you heard of epinephrine, norepinephrine? Yes? The one that I am going to ask you, because it's the one that we see in the skeletal muscle, acetylcholine. ACH is usually how it's abbreviated. So the specific neurotransmitter that crosses the synapse into a muscle, skeletal muscle, is acetylcholine. The neurotransmitter, you don't need to know the others, I was just trying to give you some familiar names. The neurotransmitter that crosses the synapse into a skeletal muscle, acetylcholine. The neurotransmitter that crosses the synapse into a skeletal muscle, acetylcholine. It is an excitatory effect. We'll talk about these much more when we get to the nervous system at the end of this semester. Some neurotransmitters are inhibitory, some are excitatory. Acetylcholine is excitatory, which means that when it crosses the synapse, its effect on the skeletal muscle is to excite it. Acetylcholine. Acetylcholine will only be released to cross the synapse when calcium is present. So we have to have the first couple steps. Nerve impulse gets to the end of the axon. That nerve impulse causes calcium channels to open, calcium comes in. Then and only then, when calcium comes in, does the end of the nerve release acetylcholine. And acetylcholine is what crosses the synapse to deliver that nerve impulse into the muscle. It diffuses across the synapse and it binds to the muscle. Isn't it amazing that all these things are happening all the time? Every muscle contraction. When you wrinkle your nose at me, or raise your eyebrow at me, or shake your head at me, these are all muscle contractions. When you leave the lab and give me the middle finger behind my back, these all we're relying on muscle for every single, you move your eye, you're writing, for every single muscle contraction. Isn't that wild? So we're beginning to explain how a muscle contracts. The very first thing every muscle needs is a nerve impulse. But it's not so easy, you see, because at the end of each nerve, there's a synapse. How are we going to get that electrical current, that nerve impulse, how are we going to get it across the synapse? A sequence of chemical events. The nerve impulse reaches the end of the axon. That triggers calcium to come in. Calcium then, when calcium comes in, it triggers this little vesicle to go to the end of the nerve. And look, it will release the neurotransmitter acetylcholine across the gap. Here is that magnified even more. neurotransmitters can be reused. So it's broken down and reused. See, if calcium didn't come down, then acetylcholine would stay in this vesicle. So if there's no nerve impulse, those vesicles just store the acetylcholine. It is only when the nerve impulse comes down, calcium comes in, these vesicles move to the end of the nerve, release acetylcholine, which crosses the synapse to deliver the nerve impulse to the muscle. I have other pictures to show you different views, but the steps are the same. Is it feeling so scary? Okay, so now we're going to move on. So here's my plan. I'd like to go through the steps of muscle contraction once, then we'll take a break. I'll give you the exams back, and then we'll go through it again. So I'm going to go through it again. You don't have to get everything down this time, because that's just the beginning. All right. So you will see that I came prepared. I also have these same steps written in a simplified form. If I go back to Blackboard, I have posted, look, muscle tissue lecture review videos. There's a link to me, I'm going to post the whole video, but I also have links to me just reviewing the anatomy. And there's a link of me reviewing what I'm about to go through right now.
Now, I've been using Grammarly for years. Now, the great thing is, Grammarly is free and easy to use. It not only helps you check your grammar, but also ensures that your tone is appropriate for the audience you're writing for. Can that make sense?
Alright, so here we are back for our second review video.
So what I'm about to go through, you're gonna have access to, okay? And here's where I also have that list. Just another way of simplifying. I hope those will be helpful. I think they will be. It's our first video. Now, if I just look here, right? The first thing that has to happen is that nerve impulse. So if we were to break it down into steps, the steps of muscle contraction. Number one step of muscle contraction is that nerve impulse getting to the muscle. Of course, there are sub-steps within that we just talked about. In order to get that nerve impulse into the muscle, calcium channels open, acetylcholine is released, acetylcholine crosses the synapse into the muscle. I'm incorporating that right now all into step number one. We've got to get that nerve impulse into the muscle. Now, what this next slide says is there's a latency period. And that's just a fancy word for pause. How about that? Latency. Pause. So after the nerve impulse gets into the muscle, there's like a millisecond pause. When you see that term, latency period, it just means that there's a brief pause right after the nerve. Basically, it pauses to let the muscle get its shit together. It's a brief pause just to let that nerve impulse get deeper into the muscle, to let it get ready. But it's like, we're talking about milliseconds. Sometimes you might see that called the excitation contraction coupling. We don't need to get that specific. If you can summarize what's on this list here, right? If you can tell me the first step of muscle contraction is the nerve impulse getting into the muscle. Make sure you can tell me how the nerve impulse gets into the muscle. And then there's a brief pause. Now we get into the meat and potatoes of the muscle contraction. Okay, here we go. We're going to break it down into approximately 8 or 9 steps, depending on how you look at it. I already said the first step. Nerve impulse travels in the neuromuscular junction, crosses into the muscle. Cool. You can see I have it written in words on the PowerPoint. I have it summarized here also. This might be a good way to summarize it. I also have a little, like, quote unquote picture here. It's kind of weird, but look, step number 1. The nerve impulse came down, those steps happened, nerve impulse traveled into the muscle. Well, now that nerve impulse travels deeper into the muscle. The nerve impulse travels deeper into the muscle. You might see AP or action potential. An action potential is just a fancy word for a nerve impulse. So when you see that phrase action potential, same thing as nerve impulse. So once the nerve impulse gets into the muscle, that nerve impulse or action potentials travels deep into the muscle. More specifically, it travels to, that nerve impulse travels to the sarcoplasmic reticulum. So nerve impulse travels to the muscle, crosses the synapse. Nerve impulse travels deeper into the muscle. More specifically, that nerve impulse or action potential travels deep into the sarcoplasmic reticulum. That is that sleeve that covered the myofibril. So the action potential or nerve impulse travels deep into the muscle to the sarcoplasmic reticulum. Well, then what's going to happen? If we can remember that the sarcoplasmic reticulum stores calcium, step 3, when the nerve impulse gets to the sarcoplasmic reticulum, it is triggered to release calcium. Step number 3, that nerve impulse or action potential, when it got to the sarcoplasmic reticulum, the sarcoplasmic reticulum releases calcium. Nerve impulse comes into the muscle. Nerve impulse travels deeper into the muscle to the sarcoplasmic reticulum. Step number 3, the sarcoplasmic reticulum will release calcium. It's like telling a story, isn't it? Well, then what happens? That calcium released from the SR, sarcoplasmic reticulum, travels deeper into the muscle, and it binds, the calcium binds to actin, type protein. But more specifically, it binds to that subunit. Step number 4, calcium binds to the subunits on actin. More specifically, that calcium binds to troponin. animations later that will hopefully help with this. Calcium binds to the subunit on actin. Once calcium binds to that subunit on actin, it causes a shift to expose the active sites. Once calcium binds to that troponin, it causes a shift which exposes the active sites. If you look more closely, look at the actin here. It almost looks like an olive, each one. Do you see that little eyeball, like little dark circle? In this picture, the active sites are open, they're exposed. See those little black thingies? But look up here. Here we don't see them. So when the muscle is at rest, the active sites on actin are not exposed. They're actually covered by this orange band. The orange band is the other protein subunit called tropomyosin. But can you just see here, I don't see that little black dot? Yeah? So when calcium binds to this yellow troponin, it shifts the orange band. Before, this orange band, tropomyosin, was covering the active sites. But it's only because calcium binded to the troponin that the tropomyosin, the orange band, shifts out of the way to expose the active sites. Calcium, step number 4. Calcium travels, binds to a subunit on actin. There is a shift to expose the active sites. Number 4. Calcium binds to the subunits, causing a shift to expose the active sites. Maybe McDonald's should, what's McDonald's logo? I'm loving it. Maybe it should be all about steps of muscle contraction. We're loving it. No? Hungry? But it's like a domino effect. One thing happens which leads to the next and leads to the next. So after calcium has bound to the subunit to cause a shift to expose the active sites, then we have step 5. Step 5 is the cross bridge. Cross bridging is when the myosin heads perk up and bind to the active sites on actin. Step number 5, cross bridging. The myosin heads bind to the active sites on actin. We haven't had any movement yet. There's no sliding. If I give you the same analogy of like at rest, the actin and myosin are kind of like this. Cross bridging just means that actin and myosin bind. The myosin heads bind to actin. There's no sliding yet, but the myosin heads bind to actin. Now they're touching. The myosin heads bind to actin. Now they're touching. Cross bridging. Myosin heads bind to the active sites on actin. And then we have the sliding. Then we have the power stroke. As you can imagine, the power stroke is the sliding. How does it happen? Well, that myosin head has stored energy in it. So after the binding, cross-bridging, then that myosin head releases its stored energy, causing the actin and myosin to slide together. Step number, what am I on? 6. 6 is the power stroke. So step 5 was the cross-bridging. Myosin head binds to actin. Step number 6, myosin head releases stored energy, causing the myofilaments to slide past each other. That is the sliding. Cool. But you see, there's the cross-bridging, and then they slide, but then the cross-bridge is released. Believe it or not. So the myosin head is released from actin. However, steps 5, 6, and 7 will repeat as long as there's calcium. Steps 5, 6, and 7 will repeat. So what it's going to look like this is a pretty good review here. I'm proud of myself. Summarize it. The first time there's a cross bridge, actin and myosin bind together, slide, and then release. But if there's still calcium coming down, it will bind again, cross bridge, power stroke, and release. It'll happen again, cross bridge, power stroke, release. Each successive time, the actin and myosin get closer and closer together. So at first, it's cross bridge, power stroke, release. Happens again, cross bridge, power stroke, release. Happens again, cross bridge, power stroke, release. So it's described as the myosin heads kind of walking their way down the actin. It's described as the myosin heads walking their way down the actin. It's described as the myosin heads walking their way down actin, because each successive time, the actin and myosin slide closer and closer together. This is because, folks, to contract a muscle, it's not just one nerve impulse. If I only send one nerve impulse, it's basically a twitch. Like if something's coming into your eyeball and you twitch. For a sustained contraction, like what most of our things are, we've got to send multiple nerve impulses. If I send multiple nerve impulses, then there's calcium continually being released. If calcium is continually released, then this cycle continues. With every time calcium comes down, opens the active sites on actin, cross-bridging, power stroke, release. Oh, there's more calcium? It'll cross-bridge, power stroke, release. That cycle ends when you no longer have nerve impulses coming down to tell the muscle to stay contracted. So whenever there's no longer nerve impulses coming down, then that cycle ends. And actin and myosin will return to their resting position, and the sarcoplasmic reticulum will reclaim its calcium for next time. So whenever there's no longer nerve impulses, there's no longer calcium, and the actin and myosin will boing back to resting position because they're elastic. And then the SR, the sarcoplasmic reticulum, will pick up the calcium and store it for next time. Let's pause there. We'll come back, we'll do it again. I hope this recorded. You didn't get an error message, did I?
Yay!